Up next is Anca, who is studying the neuropsychological features of patients with mutations in genes that regulate chromatin structure, gene transcription, and DNA repair during the development of the nervous system. The goal of her work is to introduce these rare conditions to clinicians and researchers. So with that, Anca, take it away.
Okay, well, thank you a lot for this introduction. This project is really dear to me because it was my project as part of my Bachelor of Science in Psychology, and I'll now continue it as part of my doctorate in clinical neuropsychology. And has been really an interesting project to do. Supervised by Dr. Lipe and Dr. Campo, which is a geneticist, and Dr. Lipe is a neuropsychologist. So today you're going to learn about a very rare and novel syndromes. So I'll tell you more about the neuropsychological profile of individuals with pathogenic variants or mutations in CHD3 and KAT6B genes. So first, let me tell you a bit of a story about CHD3. As you see, CHD3 mutations have been found in 2018, so this is quite recent, and a group of doctors, which my supervisor is part of, have observed a group of children that had something in common. And in fact, that something was very rare speech disorders. And these speech disorders are known to be childhood apraxia of speech. And this is the brain that has a hard time to plan and develop speech. So this was quite interesting to look at, and these doctors wanted to look more deeper into the genotype of these children, so they sequenced the genome of these children, and in fact they did find something that was not going quite well, where a mutation in the CHD3 gene, also called chromodomain helicase DNA binding 3 gene. And based on this new genetic discovery, here is how the new syndrome came to light, Snitcher's-Bloch-Campo syndrome. Yes, we kind of have an idea of the genotype, but what about the phenotype of this syndrome? And in fact, the phenotype has really some characteristic physical features, as we see here in these pictures with these individuals with CHD3 mutation. We see that they have some a prominent forehead, as well as widened eyes, and macroencephaly, which is a bigger head circumference. And now let's look more at the KAT6B gene mutations. So once again, this one, I'll tell you a bit of a story to start off. As you see here, the research on KAT6B mutation started a long time ago, in 1986, with Dr. Odo, which assessed 3 children, and in fact these children were a cousin, a brother, and a sister. And he observed something common among them, which was underdeveloped eyelids, animal problems, heart defects, but also very severe intellectual deficits. So for a long time, we weren't very certain of the genetic basis of these characteristics that were found among these children. However, in 2012, in the lab of my supervisor, they decided to analyze the genome of these children with these children with these characteristics, and they did find that there was a mutation that was common among them in the KAT6B gene, which is also called the lysine acetyltransferase 6B gene. And this was quite revolutionary because the syndrome was known based on the physical features. However, the genetic basis now has been kind of more precise. And these syndromes are the genitopatellar syndrome, as well as a variant of the Odo syndrome, the SBBISS. So when we look at the phenotype of these 2 syndromes, they also have very characteristic features. As we can see here in this picture, they do have a characteristic nose and a characteristic very prominent cheekbones, as well as maxillary problems. So now what we really aim to do is to look more into the phenotype, but into more the neuropsychological phenotype of these two syndromes, because as I have been said, we do know a very broad profile of these two syndromes. They have severe intellectual deficits and developmental delays, but we do want to create a more precise symptomology of these syndromes. But before I tell you a bit more about the process we went through, I do want to, I'm sure you're asking yourself, yes, but what's the importance of these genes? So I will first tell you that these genes are very important in the development of the nervous system of the embryo. So, as you know, every cell has inside of it DNA, also known chromatin, that is very tightly packed in chromosomes. And in order for the cell to undergo processes and to develop, this chromatin needs to be active, so it needs to become more loosened up, if we can say this way. And this tightly packed DNA it's tied around nucleosomes that are part that have histones, 8 histones, as we see here with the blue balls. So in order to become to allow the chromatin to be active, there are many enzymes that allow for regulation of these processes. And in fact, these enzymes are CHD3 and KAT6B are among these enzymes. For example, as we see here on the left, we have CHD3 that is part of the nucleosome remodeling complex. So CHD3, part of this complex, allow for the chromatin to be active with the use of ATP, of energy. On the other side, we have KAT6B that's part of another complex that is a histone modifier. So KAT6B within its complex adds an acetyl group on the chromatin, which allows it to become active by adding this particular group. So now that we know a bit more about what each gene and each protein that result from these genes do, I will tell you about our methodology. So we were able to assess children that had CHD3 mutations and 6 children with KAT6B mutation and 1 adult. We provided questionnaires to the parents on different domains. So we assessed the intelligence domain, adaptive behavior, more emotional behavior, social behavior, health, and coordination. And we see here the different standardized questionnaire that were provided to the parents in order to really assess their children on these domains. So what did we find? In fact, the first result that we found was expected was we did found extremely severe global intellectual deficits, adaptive behavior, and very poor skills in health-related quality of life and coordination. I really want to bring our attention to the first result at the bottom, which is the MSL and the MSL, MSEL, and later 3, which resulted in floor effects, and the ABAS, which resulted as well in extremely low results. I will talk more about it in the discussion part. And then, secondly, we also found something very unexpected, was that when it came to social deficits, there was a considerable variation among individuals in both groups. There were either individuals that did not show any social deficits, or some that showed a very severe autistic traits that did reach clinical significance of autistic syndrome. And finally, another unexpected result was the fact that on the CBCL, these individuals did not score in the clinical range or borderline range for the most part when it came to emotional, in the emotional domain. So they didn't seem to have any emotional issues, more particularly any anxiety. This is quite interesting when we look at the scores we got from the ABAS, which we looked at adaptive behavior, because they seem to be extremely low on adaptive behavior. So now let's discuss it a bit. And I like this because it touches upon the last presentation we heard, is the parent's perspective of bias. We were thinking that maybe this is why we get the results. Maybe parents' perspective could have been a bias, and pairing it with medical testing would provide a most accurate picture. And also, I spoke to you about the floor effects that we gathered. And in fact, this is where we think there must be, there can be a questionable specificity and sensitivity of the questionnaires. We are really questioning the clinical psychometric value of these questionnaires when it comes to this population. Because we must remember that the norms of these questionnaires are specific to neurotypicals. Therefore, we're asking ourselves, is it maybe it would be a good thing that in the future this population is tested with questionnaires that is a lot more suited to them and to their needs as well. So then, based on this, I wanted to compare, if it's not the best idea to compare it with neurotypicals, let's then compare these individuals with others that have other type of syndromes. And in fact, we did so in our lab, and we saw that individuals with CHD3 mutations seem to have a similar neuropsychological profile as individuals with Down syndrome, the trisomy 21. And when it comes to individuals with KAT6B mutation, they seem to score a lot better than individuals with, for example, Down syndrome, as well as Fragile X syndromes on the emotional and social levels. So this gives, comparing these individuals to other syndromes gives a better idea of these individuals' neuropsychological profile as well. So finally, I want to conclude this presentation with something quite dear to me, which is the implications. We do want to expand knowledge on these syndromes that are quite rare and novel, in order for the people that experience, that do have these neurodevelopmental disorders, to have a better idea of their own capabilities, but at the same time to know how to live better with their syndrome. As well, this also implicates doctors, clinicians, in order the increased knowledge would also lead to a better diagnosis rate. And in order for you to have an idea of these implications, I wanted to show you in a more visual way. So if you let me show you a bit of the challenges that research with rare disorders come into, and they both link clinical and research fields. In fact, like I just told you, there's limited knowledge of these syndromes because they're so rare and novel. I do want to give you an idea. There's only 60 cases of individuals with CHD3 mutations in the world, and there are only 90 cases of KAT6B individual in the world as well. So the limited knowledge of these syndromes also doesn't allow for an increased diagnosis rate, because the doctors have a hard time to pick up on these syndromes, on this symptomology, in order to diagnose these cases. So therefore, there are already few patients because of the the rarity of the cases, but also there's even fewer participants available for research, which creates some sort of a vicious circle. And this is where I wanna conclude my presentation, is really the importance of parents implication in the research. I have been very grateful that this research couldn't have been possible without the parents' motivation to learn more about their children's syndromes. I have been contacted and doing a lot of recruitment on their Facebook pages. They have a support network, and I'm amazed how the parents are really driving this research. And on another hand, it's really important for the clinicians and doctors to be aware, to gather as much knowledge on the syndromes in order to diagnose and then have more participants to research and expand knowledge and so forth. With this in mind, in order to benefit both clinical and research fields, but also to create better personalized interventions for these children with these neurodevelopmental syndromes. Thank you a lot. And I would also like to thank you, the network and this conference that allowed me to present. And I would also like to thank you, the NED team, and also Brain Canada team, which are my two supervisors' labs. Thank you a lot.
Oh, wonderful, Anca. Thank you for presenting that novel condition to us, and I guess starting the path of getting increased knowledge and awareness of the condition. So to the audience, again, if you have any questions, feel free to navigate to the page that is listed. We're happy to take your questions. To kick things off, Anca, one question I had for you, and it might be challenging to answer just because of the limited population that you do study, but are there any sex differences in the expressed phenotype of these mutations?
Yes, and well, that's really a good question, and it's not the first time I do have this question. However, we weren't not able to gather any conclusion on that because of, as you were saying, the limited data that we have right now. But for the moment, we did look at that, and there's no sex differences. For sure, we're basing ourselves on very limited data, but so far there's no differences. We're also looking forward to get more to look more at demographic differences. I am right now in contact with people from Colombia, from Spain, from Denmark, and Italy. Therefore, it'll be also interesting as well.
All right, wonderful. Well, best of luck, and I hope you are able to answer those questions as you collect more data. So I'm not seeing any more questions coming. So again, if anything does pop in your mind later on, feel free to send me an email. I'll be happy to put you in contact with Anca and direct your query that way.
Great, thank you.